blue blood, and biomedical safety. Horseshoe crab lysate in endotoxin detection. The horseshoe crab, an ancient marine arthropod that has existed for more than 400 million years, occupies a unique position in modern biomedical science. Although commonly referred to as a crab, species such as Limulus polyphemus are more closely related to arachnids than to true crustaceans. The extraordinary biomedical importance of horseshoe crabs arises from their copper-based blue blood and its remarkable sensitivity to bacterial endotoxins. This discovery led to the development of the Limulus Amebocyte Lysate (LAL) assay, now considered the global standard for endotoxin detection in pharmaceuticals, vaccines, injectable drugs, and implantable medical devices. The significance of endotoxin testing in medicine cannot be overstated. Endotoxins, primarily lipopolysaccharides, LPS, derived from gram-negative bacteria, can induce severe immune responses when introduced into the bloodstream. Even trace contamination in intravenous drugs or surgical implants may result in fever, septic shock, organ failure, or death. Consequently, highly sensitive detection methods are essential for ensuring product sterility and patient safety. Historical development of the LAL test. Modern endotoxin testing emerged from the pioneering work of researchers Frederick Bang and Jack Levin at the Marine Biological Laboratory in Massachusetts during the 1960s. They observed that horseshoe crab hemolymph coagulated rapidly in the presence of bacterial endotoxins. In 1968, they described a method for isolating Limulus Amebocyte Lysate from horseshoe crab blood cells, establishing the foundation of a transformative biomedical technology. Before LAL testing, pharmaceutical companies relied on the rabbit pyrogen test, which required injecting rabbits with drug samples and monitoring body temperature changes over several hours. Although effective, the procedure was labor-intensive, expensive, and comparatively insensitive. The LAL assay represented a major technological breakthrough because it enabled rapid, highly sensitive, and in vitro endotoxin detection without the use of mammals. The United States Food and Drug Administration formally approved LAL testing in 1977, after which the method became widely adopted throughout the pharmaceutical and biotechnology industries. Today, LAL-based assays are used worldwide in quality control laboratories for sterile injectable products, dialysis fluids, vaccines, and medical devices. Composition of horseshoe crab, Hemolymph and Amebocyte Lysate. Horseshoe crab blood, or hemolymph, differs significantly from vertebrate blood. Instead of iron-containing hemoglobin, oxygen transport is mediated by hemocyanin, a copper-containing respiratory protein responsible for the characteristic bright blue coloration. The primary immune cells present in the hemolymph are amebocytes, specialized hemocytes that function analogously to components of the innate immune system in higher organisms. These cells contain dense intracellular granules, rich in clotting proteins, antimicrobial peptides, and serine protease zymogens. Upon exposure to microbial components such as endotoxins, the amebocytes rapidly degranulate and initiate a coagulation cascade. The LAL reagent is prepared by lysing harvested amebocytes. The lysate contains several biologically active proteins, including factor C, factor B, proclotting enzyme, coagulogen, coagulin, and factor G. Among these, factor C is the most important endotoxin-sensitive component. Factor C is a serine protease zymogen that becomes activated upon contact with lipopolysaccharides from gram-negative bacteria. The amebocyte granules also contain antimicrobial peptides such as tachopulsins, tachystatins, tachycytins, and big defensins, which contribute to the horseshoe crab's primitive but highly effective immune defense system. Harvesting and preparation of Limulus Amebocyte Lysate. The collection of horseshoe crab hemolymph is performed under sterile conditions to prevent accidental activation of the clotting cascade. A needle is inserted near the cardiac sinus at the junction between the cephalothorax and abdomen, allowing withdrawal of approximately 50 to 150 milliliters of hemolymph from each animal. The collected blood is centrifuged to isolate the amebocytes, which are subsequently lysed to release intracellular clotting factors. The resulting lysate is purified and processed into commercial LAL reagents used in endotoxin assays. Biomedical harvesting operations typically extract up to 30% of the animal's blood volume before returning the crabs to their natural habitat. Although many individuals survive the procedure, post-bleeding mortality and physiological stress remain significant ecological concerns. Studies suggest that mortality rates may reach 15 to 30%, while surviving crabs can exhibit impaired spawning behavior, reduced mobility, and altered biological function. Mechanism of the LAL assay. The sensitivity of the LAL test derives from a highly amplified enzymatic coagulation cascade. When endotoxin molecules enter the lysate, they activate factor C, which subsequently activates factor B and the proclotting enzyme through sequential limited proteolysis. The activated clotting enzyme then converts coagulagen into coagulin, producing a gel-like clot. This reaction forms the basis of the traditional gel-clot LAL assay, in which gel formation indicates the presence of endotoxin contamination. Modern analytical versions include chromogenic and turbodimetric assays that provide quantitative measurements. In chromogenic assays, activation of the clotting enzyme hydrolyzes synthetic peptide substrates such as Bok-Loi-Gly-Arg-P-nitroanilide, releasing P-nitroaniline. The released compound is measured spectrophotometrically at 405 nanometers, enabling highly sensitive endotoxin quantification at pico. 2 nanogram levels. An additional pathway involving factor G responds to 1,3 beta-D-glucans from fungal cell walls. Because this pathway may produce false positive signals, specialized endotoxin-specific assays often include glucan-blocking reagents. The extraordinary sensitivity of the LAL assay results from enzymatic signal amplification, allowing detection of endotoxin concentrations far below the threshold capable of causing clinical symptoms in humans. Scientific and economic impact. The introduction of LAL testing revolutionized pharmaceutical manufacturing and biomedical safety. The assay dramatically improved the ability to detect microbial contamination in injectable drugs and medical devices, reducing the risk of pyrogenic reactions and improving patient outcomes. The biotechnology and pharmaceutical industries depend heavily on endotoxin testing during manufacturing and quality assurance. LAL assays are routinely employed in the production of vaccines, monoclonal antibodies, intravenous fluids, dialysis equipment, and implantable biomaterials. The rapid expansion of biologics and advanced therapeutics has further increased demand for reliable endotoxin testing systems. Economically, the LAL industry has become a multi-billion-dollar global market. Companies specializing in endotoxin detection technologies supply reagents, analytical instruments, and testing services to pharmaceutical manufacturers worldwide. The discovery also transformed biomedical regulation by establishing standardized quality control practices for sterile medical products. Conservation challenges and recombinant alternatives. Despite its medical value, the dependence on horseshoe crab blood has generated major conservation concerns. Hundreds of thousands of horseshoe crabs are captured annually, for biomedical bleeding, while additional pressure arises from habitat destruction, climate change, and harvesting for fishing bait. Declining horseshoe crab populations have ecological consequences extending beyond the species itself. Migratory shorebirds, particularly the red knot, rely heavily on horseshoe crab eggs during migration along the Atlantic coast. Population declines, therefore, affect broader coastal ecosystems. To reduce dependence on wild horseshoe crabs, researchers developed recombinant factor C, RFC, a synthetic alternative produced through recombinant DNA technology. Scientists successfully isolated the gene encoding factor C and expressed it in cultured cells, creating an endotoxin-sensitive reagent without animal harvesting. Recombinant factor C assays provide sensitivity comparable to conventional LAL testing while eliminating variability associated with biological extraction. Regulatory acceptance has gradually expanded, with the European Pharmacopoeia endorsing RFC methods and increasing adoption occurring within the pharmaceutical industry. Conservation strategies now include regulated harvesting quotas, improved bleeding practices, habitat protection, and promotion of recombinant alternatives. These efforts aim to balance biomedical necessity with long-term ecological sustainability. In conclusion, the Limulus Amebocyte Lysate assay represents one of the most important intersections between marine biology and modern medicine. The unique endotoxin-sensitive coagulation system of horseshoe crabs has enabled the development of an exceptionally sensitive diagnostic tool that protects millions of patients from potentially fatal contamination in pharmaceuticals and medical devices. However, continued dependence on wild horseshoe crab populations presents significant environmental challenges. Advances in recombinant factor C technology and sustainable conservation measures are therefore essential for ensuring both biomedical safety and preservation of these ancient marine organisms for future generations.